Hello, and welcome to our molecular biology primer design project. We have chosen to cover the process of primer design for the detection of the disease phenylketonuria. Our group members are Aaron, Faye, Firas, and Zen. This is the outline for our presentation. First, we will introduce the criteria used for primer design and explain the purpose of amplifying the gene. Then, we will cover the materials and methods we use for primer design. Third is the results of our primer design, and finally we will discuss the difficulties we face when designing our primers. Introduction. The purpose of amplifying this gene is because PKU occurs due to a mutation in the phenylalanine hydroxylase gene. So, we design our primers to bind to the normal phenylalanine hydroxylase gene. By amplifying this gene, we can identify if the gene is mutated or not. If the gene is amplified, it means the primers can bind to it and the normal phenylalanine hydroxylase gene is present. However, if the gene is not amplified, it means the primers is unable to bind to the phenylalanine hydroxylase gene sequence, and thus the gene is mutated and the patient has PKU. So here are the criteria that we use for our primer design. Number 1 is primer length. The optimal primer length of PCR primers is 18 to 22 base pairs. This length is long enough for adequate specificity, but also short enough for primers to bind easily to the template at the annealing temperature. Number 2 is primer melting temperature, or TM for short. It is the temperature where half of the double-stranded DNA will dissociate to become single-stranded. The melting temperature of primers should be between 65 degrees Celsius and 75 degrees Celsius, and within 5 degrees Celsius of each other. The third is primer annealing temperature, or TA for short. A high TA will result in an insufficient primer template hybridization, causing low yield of PCR product. A low TA may lead to non-specific products due to high number of base pair mismatches.
So the fourth rule of primer design is the GC content. So the percentage of Gs and Cs in the total bases of the primer should be 50 to 60%. The fifth rule is the GC clamp. G or C bases at the last 5 bases of the 3' end of the primer helps promote specific binding due to strong bonding of G and C bases because they have 3 hydrogen bonds. However, more than 3 G or Cs should be avoided. The sixth rule is primary-secondary structures. These structures are produced by intermolecular or intramolecular interactions and can lead to poor or no yield. These are hairpins, self-dimers. It is when the primer is homologous to itself and forms intermolecular interactions between two primers, and cross-dimer, which is intermolecular interaction between sense and anti-sense primer. We plan to amplify a 75 to 200 base pair product. The PCR product should be at least 75 base pairs long so it can be distinguished from any primers or dimers that could potentially form. So we have to avoid regions that have a chance to form secondary structures at the annealing temperature and avoid regions with long repeats of single bases. So if there are more than 4 repeats of single bases, that means it is a long repeat, so we have to avoid those. We also have to avoid regions of secondary structures as much as we can, and have a balanced distribution of GC-rich and AT-rich domains. Use regions that have a 50 to 60% GC content, and usually 3 to 4 nucleotides are added to the 5' end of the restriction enzyme in the primer for efficient cutting.
Okay, so for our materials and methods, we need to obtain the gene of interest, which is phenylalanine hydrolase. So to obtain the gene sequence, we go to PubMed and we search for the gene. Then we scroll down to FASTA, and from here we are able to obtain the entire sequence for the genes that codes for phenylalanine hydrolase. So basically we need to copy this entire sequence, which I've done already. Then we go over to Gene Runner. Then we need to paste the gene in here. After that, we need to analyze our selected gene to look for PCR primers that we can use. So to analyze the PCR primers, there are some parameters that we need to set. For example, the primer TM, the lowest should be 65 to 75. Then the CG content should be 50% to 60%. Then the primer length is already set 18 to 22. And for the product, it should be about 75 to 200. And the concentration, the GC percentage should be also 50 to 60%. So then we do a search. However, as you can see, no results, no primers are shown. This is because the TM might be a little too high, so we're going to reduce it to about 55 to 65. So we do the search. Then it shows us two different possible primers that we can use. So we need to do the analysis of the primers. So as you can see, this is the SENS strand, doesn't have any hairpin loop. It has some dimers, bulge loop, no internal loops, and yes, match lights. So most of them are all negative. So this means that all these secondary structures will not occur, so we don't really have to worry about it. And same for the anti-sense. However, for the sense strand, as you can see, the 3' end, right, it only has one G, so it doesn't have a GC clamp. So because of this, this primer is not very good, so we will not use this primer. So for the second primer that they give us, it's the same thing. There's no GC clamp, so it isn't a very good primer that we can use. So we are not going to use this. So we will lower the temperature by 1, and we will do the analysis again. So now it shows us 2 more. So if we take a look at them, as you can see, this one has the CG clamp at the back, and the dimers, the hairpin loop, it's 0 degrees, so it will not form. The dimers are negative as well. The bulge loops are negative, no internal loops, the matched sides. And for the anti-sense, even though it doesn't have a CG clamp, but it doesn't have 3, but it has 2, so it's kind of better as compared to the previous one. And as the same thing, the secondary structures they all have a negative TM, so they will not form and they will not disrupt the PCR.
As for our results, these were the 2 primer sequences that we used. Under discussions, one of the topics that we'll be discussing is the TM value. One of the challenges that our group faced was getting a negative TM value instead of a positive one. The TM value is the melting temperature of the primer. If the temperature gets higher than the melting temperature, the primer will not bind to the DNA sequence, even if the nucleotide sequence matches. We had a negative TM, which means that the primer sequence that we obtained does not have a high CG content. CG nucleotides form 3 hydrogen bonds with each other, whilst AT nucleotides form only 2 hydrogen bonds with each other. Thus, there will be less hydrogen bonds between the primer and the DNA sequence, and less energy is required to break the hydrogen bonds between them. A positive TM value will be able to ensure that the primer and the DNA sequence will be able to bind well. The next thing we'll be discussing is the primer melting temperature. Another challenge that our group faced was that the optimum temperature that we achieved for our primer sequence was 53.5 degrees Celsius. However, the required temperature for a good primer is between 65 degrees Celsius to 75 degrees Celsius. Our group tried our best to obtain the optimum results by looking through the different criteria and making changes. However, we can't seem to find a result that matches all our criteria. These are the references that we use. They have been done in Harvard Style Citation. We have come to the end of our video presentation. We hope you enjoyed it. Thank you.